Welcome back to our SC Pharma YouTube channel. Today we are going to discuss the ELISA assay technique. ELISA, that is enzyme linked immunosorbent assay. The contents of today's presentation are: we are going to see the principle, types, procedure, application, advantages and disadvantages of ELISA assay technique. We are going to discuss about the ELISA. What does ELISA means? ELISA, it is a basic assay technique which is known as enzyme linked immunosorbent assay. It is also referred as EIA, that is enzyme immunoassay. This ELISA technique is carried out to detect and measure the antibodies, hormones, peptides and proteins in the body. Antibodies, these are the blood proteins which are responds to a specific antigen in our body. It helps to examine the presence of antibodies in the body in case of certain infectious diseases. Means if we have any infectious diseases, we can detect the presence of antibodies in our body with the help of ELISA technique. Principle of ELISA technique. The basic principle of ELISA is to use an enzyme to detect the antigen antibody binding. The enzyme converts a colorless substrate chromosome product indicating the presence of antigen antibody binding. Means what is the principle of ELISA technique? In this ELISA technique uses an enzyme to detect whether the antigen antibody binding is occurred or not. And how we can determine this? By whether if the enzyme a colorless substrate, that is chromosome to a colored product, then we can say that the antigen antibody binding has been occurred. This point is types of ELISA. Basically there are four types of ELISA, that is first one is the direct ELISA, second indirect ELISA, third one is the sandwich ELISA and fourth one is the competitive ELISA. In first, that is direct ELISA, antigen is coated to the microtiter well plate, and in this type of ELISA antigens are detected. Second, indirect ELISA, here also the antigen is coated to the microtiter well plate, but in these conditions antibodies are we can detect antibodies. Third one is the sandwich ELISA, where antibodies are coated to the microtiter well plate and we can detect the antigens. Fourth one is the competitive ELISA, where microtiter well, which is antigen coated, is filled with the antigen and antibody mixture, and here also we can detect the presence of antigen. First type, that is direct ELISA. Here the antigen is immobilized on the plate and then enzyme labeled antibody binds directly to the antigen. This antigen is immobilized on the plate and then this enzyme labeled antibody binds directly to the antigen. This technique is a simple and but it has less sensitivity. Procedure of this technique: the antigen is incubated overnight in a microtiter well plate as the solid phase of the experiment. Then next step is washing step, that is unbound antigen is removed by buffer wash steps. The next step, the enzyme conjugated primary antibody, this antibody is added to each well. Subsequent buffer wash steps to remove unbound antibody. Then again washing is carried out to remove any unbound antibody. Upon the addition of substrate, conjugated enzymes produces an optical signal via colorimetric product. Next step is addition of this substrate. This signal is directly proportional to the number of bound primary antibodies and accordingly the amount of antigen present and the concentration of antigen present in the original solution. This substrate forms a colorless product into a colored product via a colorimetry, and this signal is directly proportional to the number of bound primary antibodies. Hence we can find out the amount of antigen presence in the sample. Next is the indirect ELISA. Indirect ELISA detects the presence of antibody in a sample. We can detect the presence of antibody. The antigen is attached to the wells of the microtator well plate. Here antigens are attached to the microtator well plate. A sample containing the antibodies, primary antibodies, is added to the antigen coated wells for binding with the antigen. Next step is primary antibodies. Primary antibody containing sample is added to this well plate. The free primary antibodies are washed away and the antigen antibody complex is detected by adding a secondary antibody conjugated with an enzyme that can bind with the primary antibody. Next step is the washing step to wash out any unbound antibodies, and after that we have to add the secondary antibody conjugated with an enzyme. Means this secondary antibody which is conjugated with an enzyme, this secondary antibody binds to this primary antibody. All the free secondary antibodies are washed away. Again next step is washing step, so that unbound secondary antibodies will wash away. A specific substrate is added which gives a colored product. Next step will be addition of the substrate, where the colorless substrate will formed into colored product, which means that antigen antibody binding has been occurred. The absorbance of the colored product is measured via spectrophotometry. This ELISA, indirect ELISA technique is more sensitive than the direct method. Next one is the sandwich ELISA technique. Sandwich ELISA helps to detect the presence of antigen in a sample. The microreactor well is immobilized or coated by the capture antibody. What is done? This microreactor well plate is immobilized by capture antibodies. The sample containing the antigen is added to the well and washed to remove free antigen. Next step is antigen containing sample is added and again it is washed to remove any free antigens or unbound antigens. A primary antibody which binds to another epitope on the antigen is added. Means first we have added capture antibody, then again the antigens, and again we are going to add the antibodies. Then an enzyme linked secondary antibody, that is detection antibody, targeting the FC region of the primary is added. The well is washed to remove any free secondary antibody. Then again this secondary antibody is added, which is attached to this primary antibodies. The enzyme specific substrate is added to the plate to form a colored product. The substrate is added to form uncolorless product to the colored product. It is highly specific and sensitive ELISA technique. The last type is the competitive ELISA technique. The principle, it measures the amount of antigen in a sample. In this type of ELISA, it is another version of your antigen of interest that is labeled instead of the antibody. Unlabeled antigen, that is your interest, and the labeled antigen compete for binding with the capture antibody. The color or the signal produced addition of a substrate is inversely proportional to antigen of interest in the sample. Means the color is inversely proportional to the concentration of the antigens. Procedure, competitive ELISA helps to detect the antigen concentration in a sample. The microreactor wells are coated with the antigen, which are labeled antigens. Antibodies which are incubated in a solution having the antigen, that is unlabeled antigens, are poured into it. The solution of this antigen antibody complex is added to this microreactor well plate. The well is then washed to remove any unbound antibodies. More the concentration of antigen in the sample, lesser the free antibodies will be available to interact with the labeled antigen which is coated on this well plate. The enzyme linked secondary antibody is then added to detect the number of primary antibodies present in the well. The concentration is then determined by the spectrophotometry. Sample antigens competes with antigen for binding with the antibodies. In there is an inverse relationship between signal and antigen concentration. Means what we can say, there is an inverse relationship between the signal and the antigen concentration. This is the summary of ELISA technique, that is direct ELISA, indirect ELISA, sandwich ELISA, competitive ELISA. The next point is the ELISA procedure. What are the general steps for conducting the ELISA technique or ELISA assay? First, coating with antigen or antibody is the first step. Then blocking to prevent non-specific bindings. Then addition of a sample. Then next step is addition of enzyme labeled antibody. Next step is substrate addition. Then the last step is color development and measurement of the color. The common enzymes used in the ELISA techniques are HRP, that is horseradish peroxidase, ALP, alkaline phosphatase. Substrates used are TMB, OPD, PNPP, and this signal is measured via spectrophotometry. Applications of ELISA are in the disease diagnosis, that is HIV, COVID-19, hepatitis diagnosis, hormone level detection, example insulin, HCG, allergen detection in food, research and vaccine developments. Then we are going to see the advantages and limitations. Advantages of ELISA technique: it is high sensitivity and specificity, quantitative results are obtained, and it is cost effective. Limitations of this technique are cross reactivity, requires the skilled training, skilled handling, and it is time consuming. Thank you. Like, share and subscribe for more videos. Thank you.